Hello everyone. So it's the new weekend and I'm back with the new topic today. From the feedback you have received, one of my friends suggested me the topic of the horizontal gene transfer, the phenotypic mixings, the genetic shift, drifts, reassortment. This was one of the challenging topic. It's for him during the preparation of USMLE exams. So I will try to go through the topic. Let's try to explore the topic together today. um, the objective of the today's discussion would be like be able to solve the questions like this that I have started with the question today, and we'll go through the phenotypic mixing, uh, genetic reassortment, the genetic recombination, the difference between them, and some topics about the horizontal gene transfer in the bacteria and the microorganisms, the transformation, transduction, conjugation, transfection, and and at the end will be diving in uh, the mnemonic for the segmented bacteria, segmented virus. I mean, so that would be the objective of the discussion today. So let's start with the question, provided that you haven't seen the answer below. In a virology experiment, researchers co-infect the host cells with two distinct strains of a virus. Strain X, that is the avian origin, strain Y, that is the human origin. The progeny viruses produced in the co-infected cells exhibit a novel surface protein composition incorporating elements from both strain X and strain Y. This modification allows the modified progeny viruses to infect a cell type previously resistant to both the parent strains. However, upon the successive generations, the surface protein composition reverts to that of the original strain X, and the modified progeny loses the ability to infect the new cell type. Uh, what term best characterizes the observed phenomena in which the progeny viruses initially acquire a hybrid surface protein composition but subsequently revert to the original phenotype? So the options are genetic recombination, phenotypic mixing, reassortment, genetic shift. So these sort of questions are quite common in the exams, so far I know, and these sorts of questions are also quite prevalent in the question banks like the UWorld, Emboss, the USMLE RX, and everything. However, this is not the question from either of the question banks. I prepared the question myself. So let's dive into the topic today. What is phenotypic mixing? So phenotypic mixing is the form of the interaction between two viruses, each of which holds its unique genetic material. The two particles share the code proteins, therefore each has its similar assortment of identifying the surface proteins, while having the different genetic material. In other words, the non-genetic interaction between the virus particles released from a cell that is infected with two different viruses have components from both the infective agents, but the genome from only one of them. So this means that by the term phenotype, we mean the external component of the organism. It is not the genotype, it's just the phenotype. So this resembles the structural change, but not the genetic component is not changed. So for an example, to make it more clear, here we have the virus A, here we have the virus B. It has the code protein of its own and genetic material of its own, and virus B has similarly the code protein of its own and genetic materials of its own, and they both are different. They both are distinct in their characters, both phenotypically and genotypically. So when there is the mixed infection, we can see the code protein is intermixed. Like the code protein resembles to both virus A and the virus B. However, the genetic component resemble to their parent strain. So this genetic component resembles to virus A, this genetic component resembles to virus B, this again to the virus A, and this again to the virus B. So what happens on the subsequent generations? As the subsequent generation is always determined by the genetic component of the organism. The genetic component, the future offsprings, is going to resemble their parent strain. So that means that even though the code protein was mixed in the organism, the code protein in the subsequent generation is going to resemble their parent strain, because the genetic component is not mixed in this mixed infection. So this is the type of the interaction when, like, the subsequent generations is going to resemble their parent species, of course. Give me a second. Uh, uh, so that is the main theme of the phenotypic mixing. So it's just a mixing of the phenotype. The outer coat protein might be changed for one generation. However, the subsequent generation, they are going to resemble their parent strains. So I hope I was able to make it clear. Let's go to the next topic, that is a reassortment, and this is another term that is commonly tested in the exams. Reassortment is the mixing of the genetic material. So this is different from the phenotypic mixing, because that was just the mixing for the outer coat protein, and reassortment is the mixing of the genetic material of the species into new combinations in different individuals. Several different processes contribute to the reassortment, including assortment of the chromosomes and chromosomal crossover. And this type of things we commonly see in the segmented virus like influenza. And there are processes like genetic drift, genetic shift. They are the result of the same. Um, another commonly asked topic or the term is the recombination, and the genetic recombination is the exchange of the genetic material between two different organisms, which lead to the production of offsprings with combination of the traits that differ from those found in the iron parent. So this again is the exchange of the genetic material, and the offsprings are going to be different from their parent strain, unlike phenotypic mixing. So I have compared the reassortment and recombination. Here we can see the reassortment. These both are the influenza viruses, one strain and another strain. And here we can see the genetic component is mixed between the two viruses, and the subsequent generation are going to be different than the parent strain because the genetic material is changed. If you go to the recombination, this is the combination between like two different organisms. Here we can see this is the, let's say, HIV virus, and this is the enterovirus, and we can see the crossover change or the recombination change in the genetic material of the new strain, and this is like the interaction is going to be different, and the subsequent generations are expected to be different, as we discussed earlier. Now let's move on to the new topic, the horizontal gene transfer. How the genetic material gets transferred from one generation to another generation in terms of bacteria. So it is the movement of the genetic material between two organisms. It plays a key role in the bacterial evolution, and is the primary mechanism by which the bacteria have gained antibiotic resistance and virulence, and this is commonly asked in the exams. So there are different types of horizontal gene transfer. This one is the transformation. Here we can see this is the bacteria, and this is the genetic component of the another bacteria, that is like the plasmid, and it's going to uptake the naked genetic material that is present in the surroundings by different means. This bacteria is going to uptake this plasmid into the intracellularly, and that's how the genetic component is changed. Another one is the transduction. Transduction always occurs through the viruses. Here we can see the bacteriophage virus. This is going to infect the bacteria. It gets intermixed with the bacteria. It uptakes the genetic material, and after the lysis of the bacteria, or the when it goes to invade another bacteria, then the genetic component gets changed, and this is transduction is always through the virus bacteriophage. Next one is the conjugation. We can see there is one strain, that is the donor strain. There is one receiver strain, and there is the sex pilus, through which the genetic component gets changed. And the last one is the transfection. and this is usually done in the labs using the chemical and the electrical external energies. So let's go by one to one. The transformation, like we discussed, is the uptake of the genetic material from the environment by the bacterial cells. In nature, this genetic material often comes from the adjacent lysed bacteria and can include the plasmid DNA or fragmented DNA released into the environment. So this is the component that is released in the environment, and the new bacteria, the bacteria that is near to that component is going to uptake that plasmid. That's how the genetic component of the bacteria gets changed. So in the transduction, transduction occurs when the foreign DNA or the RNA is introduced into the bacteria or the eukaryotic cells by the virus or the viral vector. If we go through this chart, the first image, we can see this is the bacteriophage. By bacteriophage, we means that the virus that attacks the bacteria. There are different types of viruses, and this one attacks the bacteria. That's how it got its name, bacteriophage. It injects its DNA inside the host bacterial cell. So bacteriophage attached to the bacterial membrane. It injects its DNA, and here we can see the bacterial chromosome. So in the second image, the phage enzyme that is located here in the cytosol breaks down the host DNA, or the phage enzymes. Sometimes the virus carries it. It is breaking the bacterial chromosome. In the third picture, we can see the cell creates new phages, including the host phase and the host DNA. So multiple bacteriophages are producing over there, and in the fourth image we can see they are developing into the new bacteriophage, and once they get released from the host cell, they are going to the next bacterial cell. This is the recipient cell, and the cycle continues. And once they inject the chromosomal material, then it's going to get incorporated into the new bacterium, and this is how the transfer of the genetic material from one bacteria to another bacteria occurs through the medium of the viruses, and that's how the transduction takes place. And there are different types of the transduction. One is the lytic ones, another is the lysogenic ones. The lytic phages hijack the bacterial host machinery to make the more viral particles. Eventually the cell lyses, releasing the newly formed viral particles that can infect another bacteria. Another type of the transduction is the lysogenic phase, in which the genetic material is incorporated into the host genome at a particular integration site, and the integrated phase remains dormant until it is triggered to enter the lytic cycle. And this is how the transduction takes place. So without much delay, let's move on to the end of the topic, that is conjugation, and it is straightforward like we discussed earlier. Here we can see the donor bacteria. It has the pilus, and the donor releases the chromosomal material to the recipient, and at the end there are two donors. So sometimes the question is asked, the end product of the conjugation, like one receiver, one donor, is both donors. So end product is both donors, not one recipient and one donor. It's going to give both donors. That is important to know. And last one is like the transfection. Transfection is the process by which the foreign DNA is deliberately introduced into the eukaryotic cells through non-viral methods, including the chemical and the physical methods in the lab. So guys, this was all about the horizontal gene transfer and the objective of today's discussion. Let's go to the explanation of the above MCQ. So in the question, as we know, there are the subsequent generations, the surface protein compositions reverse back to the original strain. That's how we'll be able to say that the genetic component was not changed in the interaction, and the answer is phenotypic mixing. So at the end, like I always do in my videos, let's go to some one-liners that the read and digest portion. The one-liner for today's are like By one-liners, I mean that these sentences they should be at the tip of your tongue, and they are commonly tested in the exams. The segmented viruses, like the influenza virus, shows reassortment. The genetic drift can cause the outbreaks or the epidemics. The genetic shift can cause the pandemics. So the genetic drift, by we mean the point mutations. There is no more, the change is minimal. However, the genetic shift, it occurs by the exchange of the whole chromosomal material in the segmented viruses, and it can result to the pandemics. So shift is the major change, drift is the minor change. That is important to know. Hepatitis C is the can cause the antigenic variation. So it is one of the example of the antigenic variation. This is important to know, that can help the hepatitis C to evade the host immune reaction. Hepatitis B and D interaction is an example of the complementation. So by complementation, the hepatitis D can't live independently, and hepatitis D is an inflammation of the liver caused by the hepatitis D virus, which requires hepatitis B for its replication. So one organism is requiring another for its replication to cause the disease. That is an important example of the complementation. This is important to know. So hepatitis D infection cannot occur in the absence of the hepatitis B virus. So I have mentioned one mnemonic at the end, like I do in most of the videos. So mnemonic for the segmented virus can be remembered with the mnemonic BOAR, BOR. So the boring viruses are segmented virus. So by B is the Bunia virus, O is the orthomyxovirus, A is the arena virus, and R is the Rio virus. And it is important to know how many segments the virus bear. So the arena, right? I have included this image from baronrocks.com. That was a really awesome mnemonic, and I really liked it to remember while my preparation of the USMLE. Arena, let's write A like this, and this is important to know. The arena viridae family has 2 segments in their genetic component. Bunia viridae has 3 components. Let's write B like this. Ortho, by ortho we mean it sounds like the octo and 8 components, and the influenza virus is one of the example that belongs to this family. And Rio viridae family has the 10 segments, and if we count the letters of the Rio viridae, it is equivalent to the 10, like the letters. So thank you so much for your patience. Thank you so much for your awesome feedbacks. If you want to contribute to the channel, you are welcome. You can mail me or text me in the same account. And in the end, whatever dreams you are pursuing, guys, just stay focused, and remember, there is no limit of time and space in this universe. Thank you for today. Please keep on providing the feedback, comments, and which next topic you would like me to discuss. Please mention in the comments or in the text portion. Thank you. Have a great weekend. Bye.